A specific messenger ligand binds to the outward-facing surface of the receptor. As a result of the ligand binding to the GPCR, a conformational change occurs, activating the GPCR. The activated GPCR, in turn, changes a GTP for the GDP on a nearby G protein. A GTP is now bound to the G protein. A subunit of the activated G protein binds to a membrane effector protein, usually an enzyme, activating it. The effector protein, now activated by the G protein, elicits a cellular response. Some other possible responses include the following. Enzymatic activity. The effector protein may be an enzyme that catalyzes a specific substrate. More specifically, the enzyme may be a protein kinase and initiate a kinase cascade. Second messenger camp. If the effector protein is the transmembrane enzyme adenylyl cyclase, the enzyme converts an ATP to a CAMP by removing two phosphate groups and binding the remaining phosphate group so that it is attached to the ribose sugar in two places instead of one. Many CAMPs are rapidly produced, generating a very strong and rapid response. The CAMP signaling pathway that activates a cytoplasmic response protein, such as an enzyme protein kinase, depending upon the cell type. The response protein initiates some specific cellular response that may be stimulatory or inhibitory. Two cases when the CAMP activates the molecules. IP3 and DAG. If the effector protein is the transmembrane enzyme phosphatase C, the enzyme cleaves a membrane phospholipid, PIP2, to generate two second messengers, IP and DAG. DAG, the lipid protein of PIP2, remains embedded in the membrane while the soluble IP3 moves to the cytoplasm. A second messenger, IP3 and DAG, initiate a variety of cellular responses depending upon the cell type. Second case, Ca2+. In many cells, IP3 triggers the transport of calcium ions across membranes. For example, in secretory cells of salivary glands, IP3 binds and activates receptor proteins in membranes of the smooth ER to release Ca2+ into the cytoplasm. Here, Ca2+ is a second messenger, triggering the release of salvia. GPCR signaling is deactivated when GTP is hydrolyzed. When the GTP that is attached to the effector protein is hydrolyzed, the G protein coupled receptor pathway is deactivated. The released GDP is free to reassociate with the G protein.